You planning to sample this, pilot?
Yep.
Should I spin up the HPU?
You may. And yeah, you can make sure to retract the drawer and retract and extend the starboard dial box pin.
Copy. Spinning up to 1800 RPM.
That's good.
Retracting drawer.
Yeah, you can come in.
Retracting port bio.
Oh, and there's another Komatsu crinoid we just went over.
I have a bit more zoom.
Excellent, thank you, pilot.
75 arms.
And video.
Yep. I was planning on putting this one starboard.
Yes, you do. Port inside has a sample.
Okay, pilot, I closed the drawer and then retracted and then extended the pins.
Okay, make sure you only extend the starboard dial box pin and make sure the port one is retracted. We don't want to let...
So this, because it's 5-armed and yellow, I have no idea whether that actually means that, but this might be some
Port
kind of hyochronid.
Starboard extended.
Thanks.
Yeah, so that's what me arms, but I just don't know how many 5-arm start crinoids are out there.
Okay, I'll get a quick stab of
What did you say, Chris? Sorry, we didn't catch that.
Pilot, I'm going to swing a little bit to the left so I can keep you in view during sampling.
Okay. Oops.
Sorry, there's quite a discussion right now, so I'll wait. So the hyochronity, if I'm not mistaken, is characterized by having 5 arms.
Yes, I agree with that.
I'll get set up to sample then.
Okay, so Dave Burdick, who is
Do you want me to raise the lower swing arms a little bit, pilot?
a scientist
Seems like
here in
it's a little bit
Guam.
uneven ground.
He's said that there's only one known stalked crinoid from the Marianas, and that is an isocrinus. And this is not that, and so this is definitely a new record of crinoid here in the Marianas. And while we're going to collect this, for those of you who just joined us, we are at the site Santa Rosa South. This is a ridge feature where we are looking for commercial fish as well as corals to help inform the Marianas management. And we are currently at a depth of 316 meters, and we have about 2 hours left in this dive.
We're all spun up?
Yep. Do you want me to take any of your cameras?
Well, we could put, if you feel like you've stopped moving and you're ready, we can put a top view from that camera, like the pilot cam is good. And then run it over. Oh, it's even aiming there. You just have to focus it.
I will point out that I think we are allowed to collect up to 6 specimens
Yeah, there you go.
per dive. So if there is any worry that we won't be able to collect any other biology after this, don't worry. We probably still can get one more. I think because the local Guam scientists are interested, as this is a new record, we should collect this one.
Definitely collecting
But
before
I
I
can
reach.
Okay, so we're definitely collecting.
No, I'm asking.
No, and I'm telling you.
Oh, okay.
Yes, pilot, we're definitely collecting.
Okay. Great. Thanks. I was going to say, that's not my call. I'm just live, reaching out. Out there? Does that look okay?
Perfect, thank you.
Want me to open the drawer?
Let me get the arm out of the way. And you can tilt up on my HD.
Copy.
Oh, not quite that far, but that's good enough. You can draw out if you'd like. Give a good view. There it is.
I think on the manips I can see pretty well.
Yep, just watch that port side rock box. Make sure we don't drag at all. And watch that manip cam. Make sure the port bio box doesn't open
Okay.
as you go. If you see any of those things happening, we'll stop and reset. You can zoom all you want video if there's anything of interest.
Okay,
It's in the
opening
dark
drawer.
Which bio box is this going into?
now. I suppose starboard inner.
Okay.
That's all right.
Thank you, pilot. Yep.
Is that open on the port?
Nope. It doesn't appear we're touching anything. So. Good. That's plenty, right? Yeah, I can get it in just like that.
Okay.
Is there any zoom on the main HD video?
Not at all.
Okay.
Full wide.
Okay. And could you bring up my starboard manip cam? Over here is starboard manip on this camera. Oh, port, this is starboard manip down below. Yeah. Okay, yeah. And then pilot or mini Zeus on this one so I can see them from above. Yeah, we can zoom out too on that. Perfect. Awesome, thanks. Why don't we try the mini Zeus, see if I get a better angle. Oh wait, I've got a good one. From the, switch this to the port minute, if you don't mind.
Sure.
Thanks. Great, thanks.
Yep. Sorry.
Are we floating?
Yeah. Looks like we are.
No, is the coral floating or the cyanide floating?
I don't think so. I think D2's floating a little bit, it seems like.
Oh, that's okay. Stay in there. Stay in there. Great. Hands clear. You can close the drawer.
Closing.
Great. Thanks.
Great work,
Closed.
pilots. Thank you very, very much.
And retract the pin.
Pin retracted.
Awesome. Thanks a lot.
Yeah.
What's the piece back to?
Thanks.
I'm taking my camera back.
Should I spin down HPU?
Yeah. And continuing on. Get the ship moving again.
HPU's off.
Thank you.
Pilot, I'm heading 55 degrees right now. I'm going to swing to the right to go back to 100.
Perfect. Just adjusting my Z bias. Keep sinking a lot.
The cryonite's pretty heavy.
So there's a red asteroid just below us. Oh, another asteroid to the right that looks pretty big. And then a multitude of crinoids and an anemone on a dead coral, as well as several different types of coral just in the frame alone right now.
That fan over, or that circle over on the left is really...
Yeah, I think that's an anemone or an actinarian.
Mind if I go look?
Sure, go for it.
Alright. Video, I want to come in push. Oh, am I looking at the back or something? Hard to tell. Want to come in partial video?
Absolutely.
Thanks. See the big circle we're looking at there?
Huh.
Funny looking guy.
Oh, no, it's not an actinarian. It's just a really
600
a crinoid with like a bajillion arms, exactly.
Hi.
So it's actually, each of its arms then branches into two further arms at the end. But still, that is a lot of arms.
That's full zoom.
So this looks more like what Asako was talking about from Japan, the metacrinus. As you can look at the stalk, you'll see it's quite short, and it's got long cirri on it.
Take a look at the stalk. Stalk seems long. Lots of feet.
Oh, it does have lots of long cirri on it. So look at that, two different stalk crinoids right next to each other.
The This one looks way cooler than the last one.
It does, definitely. I think we may have made the wrong choice. So that's 2 new records of stalked crinoids for the Marianas region in the space of 5 minutes.
Okay. That looks clear when you are video.
Okay, that's absolutely beautiful, pilot. When you're ready to move on, we are ready to go as well.
All right, I'll get a quick establishing as you.
Yeah, I'm right
Start
on
to
top
go.
of you.
All right.
Right. There's 2 of them actually.
Nice
There's one
spotting,
right
pilot.
behind it too. 2, 3 of them. 3 of them all in one spot.
Okay, some more whip corals, primnoids, a couple squat lobsters in between, paragorgids, the bright red ones.
Another stock crinoid. See the fish? Have we seen that guy yet?
No, where are you seeing a fish?
Take a partial video.
Sure.
He's moving, so I want to catch him before he goes in the crack.
Oh, yeah.
That's different. Oh, yeah. I think that's something called an odontanthias. It's related to the plectranthus. I don't recognize the color pattern,
If you'd
but
like.
I'm not that familiar with it. But it's quite colorful. You see the extended
Okay,
fin
to zoom
rays
more,
on the back part of the dorsal fin and the long filaments on caudal fin. They're really bright colors. That's very characteristic of odontanthias.
Take it.
Again, I'll have to get into the literature and figure out what species this might be. They're related to the shallow water, very colorful Pseudanthius diving a lot, big swarm, very open palau and Red Sea. And this species of this genus is much larger, but still not very big fish. Beautiful colors.
Thank you, Bruce. Yeah, it is a beautiful color. So there's loads of stalked crinoids all around this area. So I think this was a case of, you know, buying the first thing you see and then regretting it, maybe, a little bit.
All right, so let's just catch him.
But at least it's lots more visual records of crinoids for the Marianas
Take a
area.
little closer look right here. One.
Okay.
This video.
Let's see if we can get any more details.
Now that he's tired.
Attention all hands, the operations meeting will be held in the forward lounge at this time, 14:30. Operations meeting in the forward lounge at this time.
That gives a bit of information. He's found a good hiding spot.
Yeah, hide behind rock or coral. All right, continuing on, so I can keep up with the ship move here.
I wanted to check in with the shoreside scientists. I think we still have about almost 2 hours left in this dive. And I was wondering whether people thought we should still continue to our original waypoint or whether we might want to move off in a different direction. Any thoughts?
Well, I wouldn't mind moving on to the original waypoint, and the reason being is this definitely qualifies as a high-density community, and it would be pretty interesting to see how far it extends up onto the platform.
Perfect. That's exactly what I wanted to know, is whether we should continue to that waypoint or not. So that will be that's the plan we have in right now with navigation, and I think that's the plan we'll keep.
Sorry, I missed what you said, Cara.
Oh, there's a fish on the left.
Another little
You
fish
tiny
on
bit.
the left center.
Oh, running around over there.
Is there a fish on the left side?
Oh, he's about in center screen now, but looks like he's speeding away.
Now it's moved into the center. It's moving away rapidly, though. I don't know if we'll be able to catch it.
Go rapidly in that direction. That'll help our track, actually.
Did anybody see that fish? I didn't.
Come in partial. See if
Sure.
we can catch him. Hold there.
Hold.
Go a little more.
My best guess on this is a little fish called Grandma Toyota. That's the video of one of the last guys, fellow guys. It's in the family, Callahan.
I was cooking along, chasing
Yeah.
after him.
But I think it's
He's a little more agile than I am. I think I lost him. Oh, there he is. He slowed. Back in that direction he goes.
I think same fish. He's calmed down a bit. I think one of the common names for these a guapo.
Yeah, maybe wait till he's out in front, so.
Yeah, you can keep him coming.
That's a corallium, I think. If we could just stop for a second and get a good shot.
Hold there, video.
So this is one of the commercial species of coral, which we've been looking for.
And,
Sorry, I'm talking about the
Do you mind if I
shrub-like
commercial
brighter pink in the foreground with the white tips in some places.
Yep. Come on in, video.
Okay.
I think this is kind of, I don't know what's going on. This is it's an unusual corallium. Maybe Amy's still on. I'm not sure. It's pretty late. There she is.
So could it be like a hemicorallium?
Yeah, I think so. Did I say that with an abundance of confidence?
No, you didn't. And mine was definitely phrased as a question. And it's got this beautiful stalk crinoid again growing through it. What looks like a primnoid also sticking up in the background of this shot.
There. Alright. Go as you'd like. Here. Try to hold this still.
I'll go in on this edge here.
Actually, I want to revise that. I think it almost looks a little bit more like pluricorallium.
Yeah. Okay, so maybe a pluricorallium then. But definitely a species of commercially valuable coral here. And also it's got a comatulid crinoid attached to it as well.
Could we
Oh,
center
3
the
of them.
scene until
Okay.
just a touch?
Yep. 3 comatulids, 1 stalked.
Getting establishing with the that vertical coral included.
Okay, that's
too.
great, pilots, whenever you're ready to move along.
Okay. We'll just gonna get a quick establishing for
Perfect.
video, and then we'll
Yeah. Beautiful.
continue on.
All right. Thank you, Beth.
Welcome. another one of those urchins down there. There's another crash that fell in. Looks like a little bit.
So we've definitely made it into a very high density community, full of corals and full of lots of crinoids, both stalked and comatulades, and of various descriptions.
So,
Geology-wise,
no, I was actually just going to ask a biology question. So
Oh,
again,
no.
it does seem like it's, even though it's high density, it doesn't seem like it's got
as diverse.
yeah, as much diversity as where we were yesterday. Is that
Since I got to float up
reasonable?
Like this,
Is that
do you wanna just
correct?
zoom past the
I think
seats?
that is quite correct. I think some of the
Video.
species here may be similar
Sorry, brother.
colours,
Mhm.
so don't let that
Copy.
fool you. But
Could, zoom in
compared
a
to
little past
yesterday
the
where we
seats.
had
Of course.
bright
Yeah, thank you.
purples, yellows,
Right.
and they were, I mean, we had pluraxids, we had isodids, primnoids, chrysogorgids, we had everything yesterday.
See the fish.
Yes.
So this just looks
So
a
I
little
think
bit different.
I think I mean, it's definitely different to the community we were seeing yesterday. We've barely seen any
Only another
acinellas
one.
or neurellas today.
You said partial?
Yep.
Great.
Just to start seeing if we've seen this.
Sounds
It looks
good.
like something we've seen
Yeah.
a little more if you'd like. Oh, that's the stripe. Yeah.
Yeah, I think this is the same one we've seen.
Looks like another one of the Atlanteans
Yeah.
that we've been seeing. The gold bar, open bar.
Bruce, was that you? If so, could you move a little closer to your mic?
Okay, I'll try to remember to do that.
Oh, and there's
Oh, that's
another
better.
fish just to the right, upper right corner.
Yeah. Another one of those flat striped fish.
Oh, look.
At the right is Antigonia boarfish.
Well, I think we did see one of these yesterday as well, didn't we, Bruce?
Yes, actually saw several of them when we got up into the shallow depth, and I would expect to see more. They're quite abundant. Yeah.
Some more of those
There's a couple of
chrysogorgids we've...
maybe more that are possibilities. Is that the one? Yeah, Chrysogorgia?
They look like the chrysogorgids we collected yesterday. Well, chrysogorgia question mark. That we collected yesterday.
The thing that looked like a dandelion?
Exactly.
Well, the Antigonia, for a long time, was thought to have been related to the dory, some of the fishes,
I'm gonna let him go.
like the tinsel fish. But now with the genetic work that's been done, they're thought to be more closely related to surgeon fish, shallow water surgeon fish. They are to the dory. The radical change in the places of the biological fishes.
So, thank you, Bruce. Scott France has weighed in on your diversity question.
Oh.
And he said that today alone, we've had several species of primnoids, 3 different isidids or bamboo corals, 2 or 3 paragorgids or bubblegum corals, a corallium or 2, and a couple others. And so he thinks the diversity is definitely comparable to yesterday, but the color scheme is a bit more muted.
And
so
there's
it's not as
there's a
easy.
dominant species that we didn't
Yes,
really see yesterday.
Exactly.
Thank you, Scott. That's really
Yeah,
helpful.
that is extremely helpful, Scott.
This is why we make the geologist ask biology questions for clear answers.
And I think we can accommodate your request if any of the other scientists concur, Scott. We did say that we only collect 4 to 6 specimens per dive, and we like those to be either new records, new species, or one of the dominant animals in the area. And these isidids
Fit
do
that.
seem to be, one of the dominant species in this area.
Are you looking for a collection right now or no?
No, not yet.
Oh, okay.
Just continue on for a bit while we think about it.
Are you in a good position for a collection right now, or not?
Wait, but I want the other scientist. Chris, do you think we should collect one of the isidids?
There's plenty. I'll just push them along. And when you guys say...
Chris stepped out for a moment, but he'll be back.
Okay, sure. So, Scott, do you think these isidids are a new species or new records? The fact that you have Ostomyces ? Makes me think perhaps there's another one of those Echinothurides on the left, just gone out of screen, and just loads and loads of crinoids everywhere.
Ooh, look at that fish. I almost missed it. If it didn't move, I wouldn't have seen it.
He's a little different.
Yeah, come on in.
Can we zoom in on that fish? That's something different again. Let's say Cryonema. I think this is a fish called Cryonema. Cryonema. No, it's Chryseries. Cryonema Chryseries. It's a similar that's species we also have here in Hawaii. They're called duckbill fishes. And these are actually thought to be related in a different family, but same grouping as that little sandperch that we saw earlier with the black stripe on the side. He's quite curious. He's looking at the camera.
Trying to come in a little closer, and I didn't push
That's
up
good.
enough.
Ooh.
Cool. Look at his back.
I'm going to zoom since he's get a different view.
Okay.
Sounds good.
As a reminder to all the shore-based scientists, while we're watching this delicious-looking fish, we will be having our next science meeting. He turned. We'll be having our next science meeting probably about 15 minutes after we leave the bottom today. So, no, we're not?
5. I say 5.
5?
Just because we have quite a short ascent time.
Okay, so
And we
we'll
have
be doing it
samples coming
almost
up.
immediately after we come
Yeah.
off the bottom.
A quick
So-
break just to let people
Eat people.
take breaks, yeah.
And so just keep it in mind, and hopefully some of you will be able to stay on the line.
He reminds me of ice fish from the Southern Ocean. Do you know what I mean?
All right.
Okay. That's pretty neat.
Copy.
Great. Thanks. Bye, fish.
Les would like to look at that Acanella, I think, that's straight ahead. If we could take a quick look.
So pilots, if we could just look at this orange Acanella on the left. Yes, please. Acanella. Yeah, this guy right in, moving into the center.
Yeah. Just gonna zoom around and see what a better view. And a little low, so I get in the dark. Look at that a little bit. Come on in, video, when you're ready.
Zooming.
If we could also get a branch point on it, that would be great.
Copy that. Branch point. I'm gonna push down and try to find a branch point that's visible. Pretty
Okay,
hard with all
great.
the polyps. There's one. A node branching the node, it looks like. Scan down and see if there's another
Yeah,
one here.
bigger
Near
one.
the
Yeah,
base.
there's a bigger one.
After this, could we get kind of a look at what all those white things are on the bottom? There could either be corals or sponges. We're not really sure.
White things on the bottom.
Okay, sure.
We don't have much longer, so let's come out partial real quick and look for the white thing.
Okay.
Oh, I see.
Is there any chance just to zoom quite at the base of this coral first?
Yep, there's a white
Quickly.
thing right next to it. So
And then, yeah, exactly.
Perfect. Going in for a quick shot of both.
I can hold it there. Very inopportune moment.
It's full.
So what does the base of a coral tell you about identification?
Chris, you're going to have to answer that one. I know they're crucial in aging them.
Oh.
That's about all I can tell you.
So we have a consensus here that it's a branch sponge, but it's not like anything we've seen before. And we did see some branch sponges down in Johnston Monument last year that basically were believed to be in the genus Stellodorix, but this is clearly different. So I don't know how many varieties of branch sponges they have.
Does that make it worthy of collection?
Well, I'm pretty biased, but I think so. But
I think
actually ahead of us a little
then we'll have to placate the coral people too after that.
basically, because we haven't, I feel like we've neglected the coral people all day, despite them being the dominant animals around here.
We collected coral on our first specimen.
Yeah.
We could take a piece of that orange coral right next to it. That would be good.
Okay, well, so why don't we, to pilot, is there a possibility we could sit here, collect that white sponge, collect a tiny piece of that orange coral, and then move off? Or is that not possible?
The ship is on the move and ahead of us at the moment, so if you do that,
So not
we will
possible.
have to stop
Okay.
and back up. It's
No,
not that
that's-
it's not possible.
No.
It'll take
No,
time.
no, go ahead.
If we can find a white branch sponge and an orange thing next to each other, maybe 50 meters ahead of here, that would be okay. But at
We
this
will
very spot,
look.
I'm already losing.
Yeah, no, go ahead. We
Ground.
can look. These are pretty common so far, I think.
It looks like there are quite a
Or they
few
appear to be right
already.
here at least.
Yes. So I'm going to push ahead and catch up, and then hopefully we can.
So is this
cut
some
it
kind
up.
of pavement, Deb? Like a coralline pavement?
That's certainly what it looks like. I mean, it basically looks like it's a flat limestone pavement. If this were on land, that's what I would call it, a limestone pavement. In a submarine environment, I'm honestly not sure what it's typically called.
There are possibly millions of those sponges here.
What are those little?
So
Why?
those are all the little sponges I think we were just looking at.
Yeah.
Are they? Yeah.
Yep. And shadows. I see the
So,
fish.
oh,
Wow.
But I
do we
have to-
know how far we need to travel before the ship is not an issue?
Can we stop the ship here, please?
What
No,
is
I'm
that?
not. Yeah.
Oh, we're gonna stop the ship, and then I'll let
And you'll settle down.
I can settle down. Yeah. Yeah.
Okay, yeah,
Just
then
stop
just-
it, please. Yeah. Okay. For
Oh, there's
second,
a
we'll
fish
take
dead
look at
ahead.
this big fish, yeah. Go ahead, partial.
Okay.
Ooh.
That fish is
Nope,
trying to
that's the
escape.
same one we've seen a lot of.
A little bigger version of it, maybe, but all right. Let's go. Two of the same we've seen, and
Oh, yeah.
we'll march ahead a little bit more for safety's sake.
Yep, nope, that's absolutely fine.
But if we see a line where these sort of stop, we'll stop there and grab it.
So that was a rather large Plecthranthius. Looks like one of the biggest one of those being. There was also an Antigonia in the same area.
Yes.
And for the geology enthusiasts, if you look at the Sirius right now, the downward-looking camera from the two-body system, you can see that there's actually, it looks like fractures on either side of where we are right now.
Lots of momentum to stop, sorry.
Oh, I have kind of a long comment about this green eel with the black stripe.
Oh, look at that.
Well, go for it.
I
He's
think there's a
with.
possibility.
Probably settle out pretty quick though.
This could be a
What are you looking
Nyrocongrity,
at right now?
and it's very similar
Almost
to
straight down.
a Nyrocongrity.
A little bit behind.
Are you still?
I asked a number of eel
Yeah.
specialists about this, and they couldn't quite agree if this was a Nyrocongrate or if it was just a regular congrate eel. But these images are great. We saw a lot of these on the pearl dives that we did down in the Line Island. It was a complete mystery to me. I guess it's correct about this. For the first
Let me watch
record
this guy
of
if he
the,
gets,
this family
next to you.
of eels, ours from the Line Islands about 10 years ago,
eel.
and now these are the Marianas. And
far out
it's the same type of thing that they saw over in the Western Indian Ocean. A major range extension.
Wow, that's.
Said all of that, well, we'd actually have to have specimens to test all of those ideas.
Yeah, I think our ROV pilots might have a bit of a job trying to collect this one.
Can barely videotape.
Yeah. Exactly.
Yeah,
I'm
that's the problem
stationary
with
now.
the fishers.
We need to find a slow and sluggish one, Bruce.
Yeah, we'll just chase this one till he's tired, and then. All right. So my co-pilot says he's basically settled out, and I see a field ahead of us of these.
Of these little
sponges.
sponges.
So I'll jump ahead, and maybe we can find a sponge with an orange guy next to it.
Yeah, that would be absolutely great. Thank you, pilot.
That's. Just pull over this way.
Just passing over a sea star and a Okeanos. It seems that we've like moved into an area of, yeah, a sort of different assemblage here where there's a lot more sponges, and some crinoids and some other echinoderms.
I don't see.
We haven't seen these sponges before now, and so we're actually shallower than 300 meter depth. So
Maybe that's why.
maybe that's why exactly.
This is not.
That's not
That
the
is-
orange one, yeah.
So you could tilt up a little.
Copy.
Something orange. These go off to your port. We stop moving, right? You want to just sort of cheat that way a little bit for me? I'll push ahead here and see.
What's the one dead ahead to the right?
Dead ahead to the right. Eye. Looks like crinoids on another one of these pink ones.
Yeah, it doesn't look like the orange Asalas.
a small sea cucumber there in the lower left. Might be worth imaging if that was possible.
Absolutely. Yeah, if you want to settle down the image, we could just snag one of these sponge over here if you'd like. Hopefully they're not glass sponge. Okay. Come on in, video. You might get fun.
Deep sea animals.
There's a genus that has long projections like this, or pseudopodia, I think is what they're called. It's called Orphnergus, but
It's not that
very
coloration,
different
though.
looking than any of the ones I'm familiar with in Hawaii.
But Chris, all the Okeanos I've seen are just sort of a transparent white color. I've never seen them with red on them.
Okay. Do you have any more zoom?
That's full. It's just very small.
Well, I'm clear whenever you are.
Okay.
Do we want to keep looking for a sponge coral combo, or
Yes, please.
grab it while we're sitting here?
Yeah, we've still got an hour and 15 minutes, so we can keep looking for the and moving towards the waypoint, please.
Moving towards the waypoint. All right. I'll continue on then. Which way are you facing along track still?
Yes,
Okay.
100.
All right, then yeah, that's fine. Oh wait, what's this? Let's just take a quick look here at this guy and make double sure this isn't what we want. And when we're sure that's the case, we can continue. Just don't want to dial it in just to stop.
Whoa, that's a lot of crinoids on one coral. That coral's not looking particularly healthy either. Oh, and also a squat lobster coming down the top of it.
Would you mind a partial pilot?
I don't know.
Could you tilt up just a touch?
Pick up a little, sure.
There's also barnacles on it, if we could just zoom a little bit here.
Go
Yeah,
ahead.
there's like a whole different, and hydroids.
Go in video.
After this, could we take a look at that other colony that's being abused by the crinoids?
Not this really abused one in front of us. Another one?
Yeah, it's the abused one behind this one.
Okay, sure.
It looks like it's a different type of coral.
Sure. So, okay, so on this we've got a very unhealthy looking coral with loads of zoanthids, barnacles, about 10 crinoids, spot lobsters.
And maybe hydroids.
I don't actually know,
over
and
here.
also hydroids. There's just the tiniest bit of coral left right there, the purpley pinky. But apart from that, everything else is not coral. It's just a whole little community just living on this one
skeleton.
crinoid tree.
So why do crinoids like being on the coral so much? Is it
Okay,
because they're filter feeders?
I can actually answer that exactly. So as can you, as a geologist.
Just wanted to make sure.
So basically these crinoids try to get themselves up out of the benthic boundary layer as much as possible. The benthic boundary layer is the seawater sort of seafloor at an interface, and that's where friction is highest. And so there's not a lot of currents that pass through that area, or water flow is a lot slower. Essentially, currents are a lot slower. So as a result, they like to elevate themselves. Oh, and there's an enemy. They like to elevate themselves off of the seafloor and out of that sort of slower layer into an area where they can catch more food with their cirri, and then take it towards their mouths, essentially.
An additional crab.
Thank you.
But anemones, ooh, and a crab. Okay, so that's hydroids, zoanthids, coral, crinoids. What else am I forgetting? Squat lobster, crabs.
Anemones.
So I think that's about, anemones. That's 7 different animals, all in this one tiny little area. 7 different species.
All right.
Losing the light on. Okay,
Yeah.
I'll get a quick establishing. If you want to tilt up.
So, pilot, if possible, we'd like to see the coral directly behind this one.
Okay.
That's also being
Just grab me a
abused.
quick overview shot for video.
Sure, that looks great.
Yeah, and then we'll get a current in my face side video. There we go. There we go.
Alright, that's good.
Okay. We'll go back to the other sickly coral. Is this the other one you were looking for?
Yes, thank you. Oh, no, actually. I think it may have been... Chris, did you mean this one or the primnoid behind?
This is a giant basket star.
This is a giant Gorgonocephalus.
This one that you're approaching right now.
Okay, great. Yeah, so this one, pilot, thank you.
Come on in, video. Oops.
It's a little out of the light. If we could get in touch.
So here is an
It's just a
absolutely
coral. Let me
huge
see that.
basket star.
So
Oh.
it's that coral right there. I don't want to
Sounds good. Thank you.
bump into it. So I'll just come around this side.
I actually thought those were coral branches when I was asking for that one. I'm amazed. That's a big one.
That's, this is a pretty extensive basket star. If we could have the lasers on when we settle down, that would be great. Thank you.
Good thing. It's just tucked in here, so I'm just shuffling around so I can get close without bumping into anything.
So for those of you just joining us on shore, we are at 295 meters depth on the site Santa Rosa South, which is a ridge feature just south of Guam. We have about an hour left of this dive, and we're exploring this ridge feature looking for commercial fish species as well as coral species. Okay, how many crinoids are there on this dead coral?
Come out a little. Just fill the frame.
And that Gorgonocephalus is a
Or,
beast.
yeah. Take my camera if you want, co-pilot.
I'm thinking an anemone. If we...
Do you want to tilt up or down, or?
Wherever they're trying to look, I think they're trying to see the anemone
Pan
that's
just
over
to
there.
the right a bit, yeah.
So you don't have to tilt just yet. I don't know, you might want to look down
I'm
at
going
the
to zoom in for a
drill
touch
star.
and then zoom out for some partials.
So once again, just a great example of how these animals raise themselves off the seafloor into the areas of more current, and how many different types of animals
Still after that anemone?
you can get. You can get on one coral, essentially. So we've got anemones, basket stars, and who knows how many crinoids.
Oh, I'm getting.
I think two different species of
so I can't
crinoid as
look over
well.
there. Out of my range. But
So the anemone is possibly a...
Yeah, so if you look at
Go on.
I'll come out for slightly wider.
If you look at the way these yellow crinoids are holding on to the branches, this is what you call prehensile cirri. They're able to actually wrap their feet a couple loops around the stem, and that's a feature of the family
So only swing around to give you a little bit more
Thalassometridae.
tether.
In contrast, yesterday when we zoomed in on that yellow crinoid, it had much stouter, shorter, thicker cirri that couldn't do that.
Right.
And that's why we thought it might be a choritometrid instead.
Great, thank you, Chris. And yes, I definitely see what you mean.
alright. You want to come just fill the screen and you can see how many crinoids are here, and then I'll take off.
Okay, let's
That'll
count them. Okay, so starting from the base. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, at least 17 I'm getting.
Awesome. Alright.
Probably 20, because we can't see the other side.
You're clear video. I am.
So 20 quite large crinoids on this one dead coral. Thank you very, very much, pilots. That's brilliant. We can move on whenever you're ready.
Thanks. Continuing on.
Is that a tilt you want, pilot? I'll give it back to you.
No, I'm good. Yeah, I can.
Most of those sponges almost look like they're in perpendicular directions.
Yeah, there's no rhyme or reason, it seems, huh? Oh, I see what you're saying. Half of them are one way and half of them are the other. Interesting. Maybe there's two general flow directions or something here.
So Nav, if you can give me an update when you have a chance of how far we have to the waypoint and whether we have any chance of actually getting there during this dive. Okay, great, thank you. So in this particular area, it looks like the community is totally changing. Much less corals, much more sponges.
If we're trying to get there, we might do 50 or 60 meter moves.
So, Devi, I wonder if it's a temperature or oxygen phenomenon. So your temperature is up to 11 degrees Celsius already, and your oxygen's over 4. So it's perhaps, I wasn't paying attention to that when we went into the transition, but I wonder if that's part of what's going on.
Yeah, definitely. I'm not really sure what it was before, though. How much lower it was that when we were a little bit deeper. Or how these temperatures compare to what we were experiencing yesterday.
Almost looks like there might be a fish